Okay, that's time. I am sending this announcement out. And I should add it to over here under videos. Add external URL. Here's the URL. Here's a title. And hit publish. That might hit you with 2 emails. Sorry. Oh gosh. It's a lot of biological sounds here. Does this help? Is that reducing? It does sound like it. Okay, this volume might be appropriate. Let me check my video. There we are. And let's go to here. I'm going to go off monitor. Welcome back. This is what we left off with on Friday. The various wavelengths that a spec like the nanodrop and any other spec you use that can do the UV range to quantify your nucleic acids and protein. You can use it for protein as well. And to quantify your DNA. I mentioned alternative patterns of cell division, and already mentioned that, so a little look at biofilms. So as hold on a sec. Sorry. So as I mentioned, previously, we can only culture about 2% of the estimated total prokaryotic diversity out there in the world. So one of the reasons is that so many are adapted to being able to only exist within a larger community. And one form that these communities come in, of course, is the biofilm. These are highly structured communities, and they do provide selective advantage. But beyond that, there are some of these organisms that can only exist within these communities. And your mouth is an example, of course. Hundreds of species there. And of course, that we have the floating microbial mats. Oh my God, I'm so sorry. I got to take a sec here. An airplane reference here, but that's a really dated reference. Yeah. Okay. So back to biofilms here. If I can get back to my page. Here we go. So floating microbial mats, one of the characteristics that get exhibited in these large mats of cyanobacteria is that there are certain cells that are able to differentiate into heterocysts, which is the form that's able to fix nitrogen. And again, of the nitrogenases that we know everything that we know about them currently, they have to be removed from oxygen entirely because oxygen will bind irreversibly where the nitrogen is able to bind. So this structure allows this differentiation to take place. So then that resulting ammonia can be shared with the community. So streamers, these are filamentous biofilms that are in flowing water that are anchored to substrate. You've probably seen them in streams before, and you might have thought they look like plants. And some of them might have been plants, but some of them are streamers of bacterial biofilm. And then they develop mushroom-like morphology in slow or stagnant water. Here is the progression of biofilm formation. So initially you have planktonic cells that will attach to the surface. They will adhere there, and then the first colonizers will become irreversibly attached. And this can be within seconds or it can take minutes. And then they're secreting more and more of the extra polymeric substances, EPS. That's hydrated. My little notes here have it misspelled. Yay. This hydrated gel polysaccharide is what we're looking at. And then there's a period of just growth and cell division, hours to days. And we have more EPS being secreted, and we have the development of actual water channels within it. And this allows secondary colonization to occur. And then we have dispersion of microbes to a new site. So that's the basics of the biofilm. Now, one of the features of how this community structure is able to change over time is quorum sensing, which I mentioned previously. Now, if you hear any sounds there, that's the kids are all out of school today, and Alexander's decided he wanted to take up residence in my bedroom and watch some TV. So if you hear kind of a hooting sound in the background, that's just him having fun over there. I mean, his helper is here as well, and as well as my wife, who's working from home today. So I'm not just ignoring all my kids to talk to you. They are being watched. Anyway, so quorum sensing, when there's a change in the environment that induces a signaling molecule to be secreted, the most simple way for this to happen is simply to have a cell density. So you'll be secreting a particular signaling molecule all the time, and then when there's a certain concentration of that built up, it can cause a switch to occur in the metabolic characteristics of the population. Now, in gram-positive bacteria, they utilize a variety of short peptides. Here's an example of one here. Whereas inacylated homoserine lactones is utilized in gram-negative bacteria. These are autoinducers in quorum sensing, and of course that mediates a coordinated response of bacterial cells. This can be a feature of pathogenicity as well, as that quorum sensed response could be to switch over to start secreting toxins, as an example. So the R side chain here on the homoserine lactone is what is different per species, and then some are recognized by more than one species. So there's a whole lot of signaling that goes on here. Fungi. There are fungi that also engage in quorum sensing, and as I mentioned, quorum sensing is definitely involved in virulence for some pathogens. So biofilms, how does that relate to human health? Well, for one, it can actually represent something of a passive defense. So there actually can be a benefit to the biofilm, and that's in the respiratory and the intestinal tract. The detrimental version includes, of course, the plaque on our teeth, wounds, any biofilms that form in wounds, and the Pseudomonas aeruginosa in cystic fibrosis patient airways. So the pathogens in the biofilms are more resistant to antibiotics. That's the fact. The reasons why, well, the extrapolymeric substances may be protective directly. The pathogens may be inactive in the biofilm, and that comes back to the quorum sensing type of behavior. And there are other phenotypic changes due to being in the biofilm. I feel like 2 and 3 they're kind of related, kind of the same thing. Anyway, biofilms do represent a harbor for some pathogens against antibiotics. Okay, that was section 1. Moving into section 2, looking at oxygen requirements for microbial growth. So let's see, what's the visual data, minimum, maximum, obligate, all these terms we're going to go through. So anaerobic environments once dominated the world, right? And now they are less prevalent since the great oxygenation event, 2.3-ish billion years ago? But they are still common. Environments like the bog, right? So oxygen depletion down here, the water is still, and as you get down far enough, there's no oxygen essentially at all. And the rumen, so the first compartment of the cow's stomach, This is the oxygen-free incubator for methanogens and other obligate anaerobic bacteria. Your gut also should be effectively anaerobic, and that's Clostridium is able to live quite well in that environment. But anyway, there's a lot of anaerobic environments around. Yeah, there's a really interesting carbon monoxide area that a bunch of animals die in because you can't smell it, and they. But that's just. I don't need to talk about that now. Okay, here is a medium that we used it in Louisville, but we're not having y'all do it. And if I do this again with a lab, I probably will bring that in, the fluid thioglycolate. It's got some technical challenges when you use this media because you have to autoclave it really close before you do the lab for best impact. And then with the way you inoculate it, it's easy to mess up, so you don't observe this. But anyway, fluid thioglycolate. So the thioglycolate is able to basically absorb oxygen and keep it from being available. And that means any oxygen that comes into your media had to be recent. And so it forms an oxygen gradient down the length of the tube. So that's, really pretty cool. This is one way to demonstrate what different organisms, what their oxygen requirements are. Ones that you're able to grow in culture at all like this, right? So your obligate aerobes, so these are organisms that require oxygen, will only be found, so the top part of your media will be cloudy using fluid thioglycolate, because the culture will only be able to grow to a depth of the, what its minimum oxygen requirement is, which if it's an obligate aerobe, isn't very deep. Obligate anaerobes, on the other hand, are only able to grow where there is no oxygen available, and that is at the bottom of the tube. And then facultative anaerobes, which the vast majority of enterics, of the bacteria that live on an inu, that inu in particular, in your alimentary canal, are primarily all facultative anaerobes. When they have oxygen available, they're able to grow at a higher culture density, which is what this cartoon is indicating at the top of the tube. But they are able to utilize fermentation. They have other options, and there's so they're able to still metabolize and grow in low oxygen to no oxygen environments using an alternative terminal electron acceptor. And then, of course, we have our aerotolerant anaerobes, which will grow effectively the same. For them, oxygen is something that either they can ignore or they have to manage, but it's not something they use to metabolize with anything with. So they'll grow about the same density throughout the length of the tube because their growth is completely independent of oxygen concentrations. And then, of course, there's the case of the microaerophiles, which we have a very strict oxygen requirement within a certain band that's not the common concentrate atmospheric, 18 to 20% in the atmosphere. Microaerophiles need less, but they need some of it. So that's the basic categories of oxygen requirements for microorganisms. So our obligate aerobes, obligate anaerobes, facultative anaerobes, aerotolerant anaerobes, and then microaerophiles. Yeah. So the way you grow those anaerobic organisms is in an anaerobic chamber. There's a couple of ways to accomplish this. You can so you have this container, and you can flush the oxygen out using an inert gas, or okay, I don't know if we do use nitrogen for that, but you can do a nitrogen purge or flush. And if you flush the container enough times, you can effectively remove all the oxygen, right? There's an older way is to take a jar and then take those little teacup candles and put it in there on top of your plates, and so you leave the candle lit, you seal the jar, the candle should burn off effectively all the oxygen there. I tried that with my nitrogen fixers, but one of the issues with carbon monoxide is that it also can bind nitrogenase irreversibly. So that didn't really work out. exactly. But it could have been for other reasons too, because that particular organism didn't have nitrogenase anyway. That's a whole different story. There's also chemical packs that you can put in that will scavenge all the oxygen out of the available air. And then, so the anaerobic box, of course, you shouldn't be able to reach through. It should be sealed, and this allows you to handle those anaerobic cultures inside there. Do you see that movie Life? I think it was called, where they had that terrible hood, where that guy was able to just had that little vestibule thing, that little airlocky thing, but it wasn't sealed, and so it was actually not that hard for the organism to, to be in the same air as the organism, which wasn't what wound up killing him, but anyway, it's a terrible movie. All right, this is an example of an anaerobic chamber that I modified to grow my stuff in, except I used this gas injection port that I glued onto here to insert the stable isotope of nitrogen, to put nitrogen-15 into the chamber. But the same basic principle. This has a double lid system that's able to lock down, and I ensured that all this held gas in the really old-fashioned way by fermenting some stuff in there. I didn't wind up drinking that or anything. I just threw some stuff in to get fermentation to occur, so that I could see that this was able to maintain a seal against gas buildup. And it was only the amount of gas buildup from a little bit of carbonation, so it wasn't exactly extreme or anything. But yeah, that's another type of this anaerobic kind of chamber. This is with the gas pack system, I think, which I've never actually used. But okay, some examples. Excuse me. Allergy of some kind. That's awesome. So facultative anaerobes include Staphylococci, Enterobacteriaceae. Aerotolerant include Lactobacilli and Streptococci. So when we talk about the discrete requirements, we have the optimum oxygen concentration, the ideal concentration of oxygen. Remember your brackets for your concentration. In chemistry, what is that? Okay. Now, the optimal oxygen range, of course, is the minimum to maximum permissive oxygen concentration. So here is what the ulcer in the foot of a diabetic patient can look like. So dead tissue is accumulating in ulcers, provide an ideal growth environment for Clostridium perfringens. Yeah, I keep trying to add another letter in there. Clostridium perfringens, which causes gas gangrene. That's a good thing to keep in mind. Yeah, that's all dead tissue. Yeah. Okay, now for the species that are aerotolerant, well, okay, I'm going to explain this two ways. First, the ones that are obligate anaerobes do not have a method to detoxify reactive oxygen species. Here, I'll just go to that slide here. So they're not able to handle oxygen's pretty toxic overall, realistically. And if you're going to survive in an oxygen environment where your metabolism doesn't depend on it, you need to have a way to manage the oxygen and the oxygen byproducts. So this occurs through three main systems, enzyme systems here. We have the peroxidases, the superoxide dismutase, and catalase. Obligate anaerobes generally lack all three of these. The ones that are aerotolerant tend to have superoxide dismutase, but no catalase. Now, catalase, our cells have that, and if you've ever put hydrogen peroxide on an abrasion, then all that bubbling you see is the action of the enzyme catalase. This is something you'll also do in lab with, it's one of your tests for your, to help you identify your unknown organism, is the catalase test. It's really straightforward. And peroxidases, so, you know, various ways to deal with hydrogen peroxide, and then, you know, oxygen. So, right. Catalase. Yeah, so the positive result, it'd be what you're probably all familiar with. You've seen before on your own skin. And then the negative result, of course, will be no bubbling. Something else about hydrogen peroxide, while it is useful as an antiseptic, I suppose, its use does not promote wound healing. So do with that what you want, but all right, I'm tempted to look that up again because there's a paper all about it. It's been a while since I looked at it. Okay, so Jenny, the healthcare provider who saw her was concerned because of her pregnancy. And let's see, the immune system is downregulated during pregnancy, and pathogens that cross the placenta can be very dangerous for the fetus. And there's a term for that, torch pathogens. I know, okay, so suspicion of infection by Listeria monocytogenes. So the blood sample was streaked on blood auger, which you'll be able to do. Probably this week we will do that because we just got the blood auger in last week. And so you incubate those after you inoculate them for, 1 to 2 days at 37 degrees Celsius. So here's a clear zone around these colonies. And that tells you that there is a type of hemolysis going on. There's construction going on out there. I don't know if y'all can hear that in the background or not. Okay, so this is beta hemolysis. A complete clearing around the colony indicates beta hemolysis, so complete destruction of the red blood cells. And so then they also did the catalase test, and then indicate that these are catalase positive, so it can't be streptococcus. And then the gram stain shows short gram-positive bacilli. Okay, so, motility test, right. Cells from a broth culture are grown at room temperature, displaying tumbling mobility characteristics of Listeria. And I'll show you that. That's another type of test. All right, how serious is our condition and what are the treatments? Well, we'll answer that later. All right, so alpha hemolysis will result in this greenish cast area around here. Beta hemolysis will give you a clear area. This is clear enough that if you put a paper with text on it underneath, you'd be able to read the text through the clear zone here. So that's blood auger. It is differential, but it is not selective. Lots of prokaryotes love to eat blood. It's very nutritious for them. Okay, so here is a motility test. It's positive versus a negative, and I don't like the background on that negative test is not great. But here, this is a motility auger, so this isn't liquid. Yeah, but it's pretty low concentration, like an auger type substance. But when you inoculate these, you do a stab with an inoculating needle directly into the center. And then you need to make sure you go in and then straight back out. And then from that line, when you see this cloudy area around here, you don't really see the initial stab anymore, but you just see this area around here. That indicates motility. The inoculating organism was able to migrate out from the stab. And then here, I can't really see what's going on there, but ideally, is this supposed to be the stab line? And the whole thing's clear, but that really needed a different background on that to make that more obvious. Okay, so again, the blood auger and motility test. And that brings us to section 3, looking at the effects of pH on microbial growth. So time, 23 minutes. We're doing fine. Okay, the effects of pH on microbial growth. So minimum, optimum, maximum pH, different categories, and then give examples. So this is where we get into the some of the preservation methods that people figured out long before we understood what microorganisms were at all. So lactic acid bacteria, they are able to ferment milk into yogurt, or, you know, we can transform vegetables into pickles. Well, I mean, it's cucumbers into pickles. Is there anything else that you would call a pickle that didn't start from a cucumber? I don't know. So these bacteria thrive at a pH close to 4, so slightly acidic. And so sauerkraut and pico de gallo owe their tangy flavor to their acidity. So anyway, this acidity reduces food spoilage. When you get down to the acetic acid, you know, range, your 4.0-ish area, a lot of the mesophiles that live in humans are inhibited by pHs that go even just that low. Okay, so these are the pH ranges that I already kind of showed you here. The acidophiles. Alexander! All right, hold on a sec. You can see the dog. Oh, here we are. I can see this. Hey, Bruce, come here. Up, up. I know we tell you to not get up. Wait, look. Oh, what's that? Oh, Bruce, look. You see the Bruce? No, you don't. Yeah, okay, Xander. All right, you gotta go. But you've been outside. Hopefully you weren't out there just digging under the fence again. All right, you need to go. Okay, be right. On. What? What do you want? You want attention too?
Dude.
Hi. You look at the camera.
Good.
You gonna say hi?
Hey.
No, look over here. Say hi.
Hey.
Can you wave?
Yeah.
Wave. Say hi. Okay. All right. I'm gonna mute you. Okay, back to pH values and growth characteristics. Okay, here we go. So acidophiles down to a pH of 1 up to about 5.5. Neutrophiles cover the range around pH, around 7, around the neutral pH, plus or minus 1.5. And then alkalophiles start about 7.5 and go up to close to 12. So most our prokaryotes that we have characterized extensively are neutrophiles. And so let's see, an example of an acidophile, though, would be the sulfolobus. That's an extreme acidophile. And that the Archean ferroplasma, which is an acid mine drainage, that goes down to 0. Lactobacillus in the vagina as a normal flora constituent, and it's at a range of like 3.5 to 6.8. And it contributes to the maintenance of the pH of being about 4 in the vagina, which occurs most of the time outside of menstruation. Did you see that about the birth control pill, that the range, the allowed bleeding period is completely unnecessary? I can mention that in class, but yeah, apparently that's the thing. Or maybe I'll look that paper up and send it out as an announcement. Okay, so the adaptations required to survive at a lower pH is the increased negative surface, so the net negative charge on the on proteins, active proton pumping, of course, and then changes in the phospholipid composition. Now, alkalophiles include Vibrio cholera at 8 to 11. Oh, here we go. The bright pink Archean Natronobacterium found in the soda legs of the African Rift Valley apparently are the ones that hold the record. That's the pink pigmentation here. Oh, I just said that. Sorry, the bright pink Archean. So that's the one that holds the, we would say holds the record at 10.5 pH. Okay, and so the adaptations to allow living at a high pH like that, modified lipids and proteins, compensatory mechanisms like Bacillus firmus uses a sodium gradient versus a proton gradient to, so it's a sodium gradient proton motive force. Instead of a proton motive force, it's a sodium motive force, as an example. Okay, now, Helicobacter pylori, up until the 1980s, we did not identify that as the causative agent of stomach ulcers, of peptic ulcers. And so the recommendations for treatment were bland foods, antacids, avoid stress, and it didn't really work. So Barry Marshall and Robin Warren discovered that it was Helicobacter pylori that was causing it, and that earned them the Nobel Prize in Medicine in 2005. I gotta check something really quick because I can't remember that beeping. It's getting Poe-ish at this point. Edgar Allan Poe-ish. Let's see. Wait, who drank the Helicobacter pylori? Oh, Barry Marshall. Yeah, Barry Marshall. So it reminds me of Land of the Lost. Yeah, he's the one that did it. So they weren't allowed to experiment with that to, do Koch's postulates where you have this isolate, so you infect an experimental animal, and then you observe that the disease progresses in that animal. They didn't. They weren't able to move forward with that, so Barry Marshall just drank it. Oh, here we go. However, it was discovered. Sorry, that's just a little story from Wikipedia, I guess. In 1982, Marshall and Warren got funding for 1 year research. The first 30 out of 100 samples showed no support for their hypothesis. However, it was discovered that the lab technicians had been throwing out the cultures after 2 days. This was standard practice for throat swabs where other organisms in the mouth rendered cultures not usable after 2 days. Due to other hospital work, the lab technicians did not have time to immediately throw out the 31st test on the second day, and so it stayed from Thursday through Monday. In this sample, they discovered the presence of Helicobacter pylori. It turns out that Helicobacter pylori grow more slowly than 2 days, and also that the stomach cultures are not contaminated by other organisms. That makes sense. The pH, you're looking at 2-ish, right? So after failed attempts to infect piglets in 1984, Marshall, after having a baseline endoscopy done, drank a broth, and expecting to develop an ulcer. So 3 days later, he was developing an ulcer. So basically, he used antibiotics to do that. So anyway, yeah, that was published in 1985. That experiment was. So he took it himself and demonstrated that Helicobacter pylori did indeed cause stomach ulcers. All right, temperature and microbial growth. We're setting that here. Okay. All right, so, oh, back to Jenny with the presence of listeria. So listeriosis has a 20% mortality rate and is a risk to Jenny's fetus. Negative results on the culture from the amniotic fluid. And so they did a 16S ribosomal RNA amplification. So you got the sequence for the 16S ribosomal RNA of the bacteria. And this confirmed that there were no bacteria that had crossed. But they couldn't find the 16S sequence in the amniotic fluid, which is good news. So she got a high dose of 2 antibiotics, so multi-antibiotic therapy, which is probably something that should be considered more frequently. Ampicillin or penicillin G with an aminoglycoside like streptomycin. So let's, oops. All right, tolerant. So it's halotolerant, up to 10% sodium chloride solutions. That's what makes it a frequent source of food poisoning. Because listeria can infect animals, it often contaminates foods such as meat, fish, and dairy products. Contamination of commercial foods can often be traced to persistent biofilms that form on manufacturing equipment that is not sufficiently cleaned. Listeria infection is relatively common among pregnant women because of elevated levels of progesterone downregulate the immune system, making them more vulnerable to infection. The pathogen can cross the placenta and infect the fetus, often resulting in miscarriage, stillbirth, or fatal neonatal infection. You hear the cat? Oh my God. She's just going to keep meowing until I open the door. Oh my God, what? What do you want? What do you want? Ugh. What, you want to say hi? Oh, okay. Anyway, the other 2 kids you'll probably see during their spring break, which is different from Murray's spring break. So they'll be down at Murray probably for a few days. Yeah, that's right. You'll be happy for that, won't you? Oh, of course, now you're staring at the door. Sorry, cat. The door's great. Gonna stay closed. Anyway, this is why pregnant women, if you're pregnant, you'll generally be advised to avoid soft cheeses, refrigerated cold cut, smoked seafood, unpasteurized dairy product. Now you're scratching at the door to go back out. Okay. So, yeah. So temperature ranges. We have our psychrophiles, which are the ones responsible for food spoilage in your refrigerator. So psychrophiles grow optimally at about 15 degrees Celsius. They can also grow at 0 degrees Celsius. Psychotrophs. All right, I've got the other door closed. Maybe I can get by. I think I can get by without the other one. You're not getting in my lap right now, cat. Psychotrophs, cool, so 4 to 25 degrees Celsius. Mesophiles covers the range between 20 and 45 degrees Celsius. Thermophiles, 50 and above, or to about 80. And then once you get to organisms that can survive beyond boiling temperature, we're into the hyperthermophiles. Obviously, these are going to be at high pressure and or with a very high concentration of solutes in your solution, a boiling point elevation effect. That's right, kitty. Oh, I did this, didn't I? Look at that. So mesophiles does include the normal flora and pathogens, obviously, because our body temperature is roughly 37 degrees Celsius. Thermophiles include Thermus aquaticus. This is where we get the TAC polymerase. The TAQ is from Thermus aquaticus. Geobacillus is another example. And the black smoker, so when we talk about microbes that can survive at temperatures greater than 100 degrees C, Pyrobolus, yes, Pyrobolus. Pyrodictium archaea, they'll survive the autoclave. And one of the reasons is because they have a high GC content in their genome. Their polymerase, well, the TAC polymerase itself does, so Thermus aquaticus polymerase, has additional bonds to stabilize its structure. It has more saturated lipids in the membrane to survive, to have a membrane at a higher temperature. So these black smokers right at the vent itself can be 340 degrees. And so this is the energy input into these very local... What? Oh my God, fine. Here. Okay. Oh, yeah. Is that... You need some scratching. Okay, fine. So here's a temperature range. So psychrophiles, so cold-loving, are down here. They can go a little bit below freezing. Again, that's going to be like in high salt conditions or whatever. Freezing point depression, the other side of that equation. And the mesophiles, thermophiles, hyperthermophiles. You want to have a good idea of what's in these ranges. These aren't like literal, like hardly defined areas. These are rough definitions. But anyway, the optimal growth rate is what you're looking at for what really will put it within these ranges. Now, a lot of a lot of fungi that you might grow or work with, you work with any yeast-like stuff, that's 25-ish is where they do really well at. Okay, so algal blooms. I'm going to take that on. Oh, yeah. Okay, so that's like I have like 10 minutes left. I might take this on at a different point. So, do you remember that instance in Florida? Or that, well, not just an instance, but in Florida, they had that issue as well. You had the algal blooms, and then we also had the toxic algae as well. So you have the green algae, and so you have two things that can happen with eutrophication. Is this process where excess fertilizer runs into a waterway and causes a dead zone to be created, and essentially that process results in like an algal bloom that will scavenge all the oxygen from the water. So that's what kills all the fish and crustacea and whatnot that are in that water because the oxygen's all gone. But you can also have the specific species that will generate a toxic algal bloom, and where you have, on top of the complication of the depleted oxygen, you have a neurotoxin that will like accumulate in any of the shellfish that survive. Pacific Northwest has this problem. We have dead zones all around the U.S., and I think I have another chapter where I talk about that in more detail. But that's the ethical question in this particular Sigma Xi segment, is how do we balance that? So the damage to the environment due to fertilizer runoff with the need for improved food production. And I guess the answer is going to be found in sustainable farming practices. And there's other things you can do to help retain some of that. The problem is that the ammonia, so if you add your ammonia to a field, there's enough biota there to convert much of that to nitrates, which are much more mobile even than the ammonia is. And the nitrates are able to, go like poop through a goose, straight into your waterways. And even that doesn't get absorbed by plants, and stuff that you sprayed it on. So anyway, better farm management practices, sustainable farming practices, and whatnot. But even if you try to say limit it to organic fertilizer, much of that organic fertilizer, being poop, is derived initially from synthetic ammonia that was used on a non-organic farming environment. But anyway, Section 5, other environmental conditions that affect growth. I think we're going to get through this, despite everything else. Okay. Let's see. So some other things. Osmosis, so pressure, so chemical pressure, physical pressure, and light. So halophiles, salt-loving organisms. The barophiles, so bacteria that grow in the ocean floor, are the ones that grow around those black smokers. Pretty much have to also be barophiles. And then light, so photoautotrophs being an example of an organism depending on light. So some examples of the halophiles. So we have the red algae, Dunaliella salina, and of course the halobacterium. Ones that are halotolerant include Staphylococcus aureus on your skin. That's the mannitol salt auger, helps us say that. That's supposed to be the selective criteria there. The Bacillus cereus, Vibrio cholera, these are all halotolerant. The barophiles are largely unknown. There was one lab at University of Washington I looked at like 7 or 8 years ago that have these high pressure chambers that they're trying to look at help enumerate deep ocean bacteria. But obviously that hasn't... Dude, no, that's my headset. Leave it alone. So barophiles are largely unknown as a group. And of course our phototrophs, photoautotrophs, photoheterotrophs, cyanobacteria, our green sulfur bacteria, purple non-sulfur bacteria, and then some example of the pigments they use. Okay, so wait, sorry, I just completely ignored this. Great Salt Lake in Utah. The purple is from this algae and halobacterium. Media used for bacterial growth. Let me think about this. Do I want to go over this right now? No, I don't. That's a lot of... Oh, that was a funny story, though. This was. I guess I'll save this. Only got like 4 minutes. So, I guess that's it. I will see you on Wednesday is the plan. All right. Bye. And slideshow. Oops. I need a better button. I need an extra button to play me off. All right. See you on Wednesday. Okay.